The genetic history of Europe is complicated because European populations have a complicated demographic history, including many successive periods of population growth. The history must be inferred from the patterns of genetic diversity across continents and time. The primary data come from sequences of mitochondrial, Y-chromosome, and autosomal DNA from modern populations and, if available, from ancient DNA. History and limitation of genetic studies. Classical genetic markers. One of the first scholars to perform genetic studies was Luigi Luca Cavalli-Sforza. He used classical genetic markers to analyze DNA by proxy. This method studies differences in the frequencies of particular allelic traits, namely polymorphisms from proteins found within human blood. Subsequently, his team calculated genetic distance between populations, based on the principle that two populations that share similar frequencies of a trait are more closely related than populations that have more divergent frequencies of the trait. From this, he constructed phylogenetic trees that showed genetic distances diagrammatically. His team also performed principal component analyses, which is good at analyzing multivariate data with minimal loss of information. The information that is lost can be partly restored by generating a second principal component, and so on. In turn, the information from each individual principal component can be presented graphically in synthetic maps. These maps show peaks and troughs, which represent populations whose gene frequencies take extreme values compared to others in the studied area. Peaks and troughs usually connected by smooth gradients are called clines. Genetic clines can be generated by adaptation to environment, continuous gene flow between two initially different populations, or a demographic expansion into a scarcely populated environment, with little initial admixture with existing populations. Cavalli-Sforza connected these gradients with postulated prehistoric population movements, based on archaeological and linguistic theories. However, given that the time depths of such patterns are not known, associating them with particular demographic events is usually speculative. Direct DNA analysis. Studies using direct DNA analysis are now abundant and may use mitochondrial DNA, the non-recombining portion of the Y chromosome, or even autosomal DNA. mtDNA and NRY-DNA share some similar features, which have made them particularly useful in genetic anthropology. These properties include the direct, unaltered inheritance of mtDNA and NRY-DNA from mother to offspring and father to son, respectively, without the scrambling effects of genetic recombination. We also presume that these genetic loci are not affected by natural selection and that the major process responsible for changes in base pairs has been mutation. The smaller effective population size of the NRY and mtDNA enhances the consequences of drift and founder effect, relative to the autosomes, making NRY and mtDNA variation a potentially sensitive index of population composition. These biologically plausible assumptions are not concrete. Rosser suggests that climatic conditions may affect the fertility of certain lineages. The underlying mutation rate used by the geneticists is more questionable. They often use different mutation rates, and studies frequently arrive at vastly different conclusions. NRY and mtDNA may be so susceptible to drift that some ancient patterns may have become obscured. Another assumption is that population genealogies are approximated by allele genealogies. Barbigiani points out that this only holds if population groups develop from a genetically monomorphic set of founders. Barbigiani argues that there is no reason to believe that Europe was colonized by monomorphic populations. This would result in an overestimation of haplogroup age, thus falsely extending the demographic history of Europe into the late Paleolithic rather than the Neolithic era. Greater certainty about chronology may be obtained from studies of ancient DNA, but so far these have been comparatively few. Whereas Y-DNA and mtDNA haplogroups represent but a small component of a persona unregistered trademark sDNA pool, autosomal DNA has the advantage of containing hundreds of thousands of examinable genetic loci, thus giving a more complete picture of genetic composition. Descent relationships can only to be determined on a statistical basis, because autosomal DNA undergoes recombination. A single chromosome can record a history for each gene. Autosomal studies are much more reliable for showing the relationships between existing populations, but do not offer the possibilities for unraveling their histories in the same way as mtDNA and NRY-DNA studies promise, despite their many complications. Genetic studies operate on numerous assumptions and suffer from methodological limitations, such as selection bias and confounding phenomena like genetic drift. Foundation and bottleneck effects cause large errors, particularly in haplogroup studies. No matter how accurate the methodology, conclusions derived from such studies are compiled on the basis of how the author envisages their data fits with established archaeological or linguistic theories. Genetic studies of Cavalli-Sforza equals relation between Europeans and other populations equals. According to Cavalli-Sforza's work, all non-African populations are more closely related to each other than to Africans. Supporting the hypothesis that all non-Africans descend from a single old African population. The genetic distance from Africa to Europe was found to be shorter than the genetic distance from Africa to East Asia, and much shorter than that from Africa to Australia. He explains. Both Africans and Asians contributed to the settlement of Europe, which began about 40,000 years ago. It seems very reasonable to assume that both continents nearest to Europe contributed to its settlement, even if perhaps at different times and maybe repeatedly. It is reassuring that the analysis of other markers also consistently gives the same results in this case. Moreover, a specific evolutionary model tested, that is, that Europe is formed by contributions from Asia and Africa, fits the distance matrix perfectly. In this simplified model, the migrations postulated to have populated Europe are estimated to have occurred at an early date, but it is impossible to distinguish, on the basis of these data, this model from that of several migrations at different times. The overall contributions from Asia and Africa were estimated to be around 2/3 and 1/3, respectively. This particular model used an out-of-Africa migration 100,000 years ago, which separated Africans from non-Africans, followed by a single admixture event 30,000 years ago leading to the formulation of the European population. The admixture event consisted of a source population that was 35% African and 65% East Asian. However, the study notes that a more realistic scenario would include several admixture events occurring over a sustained period. In particular, they cite the spread of farming from a source population in West Asia 5,000 to 9,000 years ago that may have played a role in the genetic relatedness of Africans and Europeans, since West Asia is sandwiched in between Africa and Central Asia. The model assumed an out-of-Africa migration 100 kr and a single admixture event 30 kr. However, most contemporary studies have more recent dates that place the out-of-Africa migration 50-70 kr. The study also involved a direct comparison between sub-Saharan Africans and Europeans. North African populations were omitted from the study. These considerations might help explain the apparent short genetic distance between Europeans and Africans. European population substructure equals, geneticists have found that Europe is relatively genetically homogeneous, but distinct subpopulation patterns of various types of genetic markers have been found, particularly along a southeast-northwest cline. For example, Cavalli-Sforza Euro unregistered trademark S principal component analyses revealed 5 major clinal patterns throughout Europe, and similar patterns have continued to be found in more recent studies. A cline of genes with highest frequencies in the Middle East, spreading to lowest levels northwest. Cavalli-Sforza originally described this as faithfully reflecting the spread of agriculture in Neolithic times. This has been the general tendency and interpretation of all genes with this pattern. A cline of genes with highest frequencies among Finnish and Sami in the extreme northeast, and spreading to lowest frequencies in the southwest. A cline of genes with highest frequencies in the area of the lower Don and Volga rivers in southern Russia, and spreading to lowest frequencies in Spain, southern Italy, Greece and the areas inhabited by Sami speakers in the extreme north of Scandinavia. Cavalli-Sforza associated this with the spread of Indo-European languages, which he links in turn to a secondary expansion after the spread of agriculture, associated with animal grazing. A cline of genes with highest frequencies in the Balkans and southern Italy, spreading to lowest levels in Britain in the Basque Country. Cavalli-Sforza associates this with the Greek expansion, which reached its peak in historical times around 1500 BC but which certainly began earlier. A cline of genes with highest frequencies in the Basque Country, and lower levels beyond the area of Iberia and southern France. In perhaps the most well-known conclusion from Cavalli-Sforza, this weakest of the 5 patterns was described as isolated remnants of the pre-Neolithic population of Europe, who at least partially withstood the expansion of the cultivators. It corresponds roughly to the geographical spread of rhesus-negative blood types. In particular, the conclusion that the Basques are a genetic isolate has become widely discussed, but also a controversial conclusion. He also created a phylogenetic tree to analyze the internal relationships among Europeans. He found 4 major outliers: Basques, Lapps, Finns and Icelanders. A result he attributed to their relative isolation. Greeks and Yugoslavs represented a second group of less extreme outliers. The remaining populations clustered into several groups: Celtic, Germanic, Southwestern Europeans, Scandinavians, and Eastern Europeans. Genetic studies after Cavalli-Sforza, new technologies have allowed for DNA haplotypes to be studied directly with increasing speed and accuracy, giving more refined data than was available in the original studies of Cavalli-Sforza. Human Y-chromosome DNA haplogroups . There are 3 big Y-chromosome DNA haplogroups that account for most of Europe's patrilineal descent. Haplogroup A1b is common all over Europe but especially common in Western Europe. Nearly all of this A1b in Europe is in the form of the A1b1A2 subclade, specifically within the RL23 subclade, whereas A1b found in Central Asia, Western Asia and Africa tends to be in other clades. It has also been pointed out that outlier types are present in Europe and are particularly notable in some areas such as Sardinia and Armenia. Haplogroup A1b frequencies vary from highs in Western Europe in a steadily decreasing cline with growing distance from the Atlantic, 80 to 90%. In southeastern Europe, A1b drops behind A1A in the area in and around Hungary and Serbia but is more common both to the south and north of this region. A1b in Western Europe is dominated by at least 2 subclades, RU106, which is distributed from the east side of the Rhine into northern and central Europe, and RP312, which is most common west of the Rhine, including the British Isles. Some have posited that this haplogroup's presence in Europe dates back to the LGM, while others link it to the spread of the Centum branch of the Indo-European languages. Haplogroup I is found in the form of various subclades throughout Europe and is found at highest frequencies in Bosnia and Herzegovina 65%, Croatia, Norway, Denmark, Sardinia, Serbia, Sweden, parts of Germany, Romania, Moldova and other countries in the Balkan Peninsula and Scandinavia. This clade is found at its highest expression by far in Europe and may have been there since before the LGM. Haplogroup A1A, almost entirely in the A1A1A subclade, is prevalent in much of Eastern and Central Europe. For example, there is a sharp increase in R1A1 and decrease in R1B1B2 as one goes east from Germany to Poland. It also has a substantial presence in Scandinavia. In the Baltic countries A1A frequencies decrease from Lithuania to Estonia. Many people link this haplogroup to the spread of the Indo-European languages in Europe, while some limit this connection to the Satan branch of said language family. Putting aside small enclaves, there are also several haplogroups apart from the above 3 that are most common in certain areas of Europe. Haplogroup N is common only in the northeast of Europe and in the form of its N1C1 subclade reaches frequencies of approximately 60% among Finns and approximately 40% among Lithuanians. This clade is also found far into the east in Siberia, Japan and China. Haplogroup E1B1B1, mainly in the form of its E1B1B1A2 subclade, reaches frequencies above 40% around the area of Kosovo. This clade is thought to have arrived in Europe from Western Asia either in the later Mesolithic, or the Neolithic. Haplogroup J2, in various subclades, is found in levels of around 15 to 30% in parts of the Balkans and Italy and is common all over Europe and especially the Mediterranean basin. Fourth strand, on November 16, 2015, in a study published in the journal Nature Communications, geneticists announced that they had found a new fourth ancestral tribe, or strand, which had contributed to the modern European gene pool. They analyzed genomes from 2 hunter-gatherers from Georgia which were 13,300 and 9,700 years old, and found that these Caucasus hunters were probably the source of the farmer-like DNA in the Yamnaya. The Yamnaya were herders who entered Europe around 3,000 years ago from the eastern steppe region in present-day Ukraine and Russia. They may have brought Indo-European languages with them. The Yamnaya altered the gene pools of northern and central Europe. Some populations such as Norwegians, owe around 50% of their ancestry to this group. According to co-author Dr. Andrea Manica of the University of Cambridge, the question of where the Yamnaya come from has been something of a mystery up to now. We can now answer that as we've found that their genetic makeup is a mix of Eastern European hunter-gatherers and a population from this pocket of Caucasus hunter-gatherers who weathered much of the last ice age in apparent isolation. Human mitochondrial DNA haplogroups . There have been a number of studies about the mitochondrial DNA haplogroups in Europe. In contrast to Y-DNA haplogroups, mtDNA haplogroups did not show as much geographical patterning, but were more evenly ubiquitous. Apart from the outlying Sami, all Europeans are characterized by the predominance of haplogroups H, U and T. The lack of observable geographic structuring of mtDNA may be due to socio-cultural factors, namely the phenomena of polygyny and patrilocality. According to the University of Oulu Library in Finland, classical polymorphic markers have suggested that Europe is a genetically homogeneous continent with a few outliers such as the Sami, Sardinians, Icelanders and Basques. The analysis of mtDNA sequences has also shown a high degree of homogeneity among European populations, and the genetic distances have been found to be much smaller than between populations on other continents, especially Africa. The mtDNA haplogroups of Europeans are surveyed by using a combination of data from RFLP analysis of the coding region and sequencing of the hypervariable segment I. About 99% of European mtDNAs fall into one of 10 haplogroups, H, I, J, K, M, T, U, V, W or X. Each of these is defined by certain relatively ancient and stable polymorphic sites located in the coding region. Haplogroup H, which is defined by the absence of an ALUI site at BP7025, is the most prevalent, comprising half of all Europeans. 6 of the European haplogroups are essentially confined to European populations, and probably originated after the ancestral Caucasoids became genetically separated from the ancestors of the modern Africans and Asians. Genetic studies suggest some maternal gene flow to Eastern Europe from Eastern Asia or Southern Siberia 13,000–6,600 years BP. Analysis of Neolithic skeletons in the Great Hungarian Plain found a high frequency of Eastern Asian mtDNA haplogroups, some of which survive in modern Eastern European populations. Maternal gene flow to Europe from sub-Saharan Africa began as early as 11,000 years BP, although the majority of lineages, approximately 65%, are estimated to have arrived more recently, including during the Romanization period, the Arab conquests of southern Europe, and during the Atlantic slave trade. Ancient DNA The genetic history of Europe has mostly been reconstructed from the modern populations of Europe, assuming genetic continuity. This is because it is far easier to retrieve DNA from living subjects than ancient human remains. However, a small number of ancient mtDNA and Y-DNA analyses are available from both the historical and prehistoric periods. In 2015, researchers reported on the DNA analysis of 94 skeletons mostly 8,000–3,000 years old from Europe and Russia. They identified 3 major waves of human migrations into Europe: the original hunter-gatherers, Neolithic farmers from West Asia about 8,000 years ago, and a third wave about 4,500 years ago from the Pontic-Caspian steppe region of the Yamna culture people. Relation between Europeans and other populations equals, a 2007 study by Bourque, which utilized about 10,000 autosomal DNA SNPs, arrived at similar results. Principal component analysis clearly identified 4 widely dispersed groupings, corresponding to Africa, Europe, Central Asia and South Asia. PC1 separated Africans from the other populations, PC2 divided Asians from Europeans and Africans, whilst PC3 split Central Asians apart from South Asians. European population substructure equals, a study in May 2009 of 19 populations from Europe using 270,000 SNPs highlighted the genetic diversity of European populations corresponding to the northwest to southeast gradient and distinguished 4 several distinct regions within Europe, Finland, showing the greatest distance to the rest of Europeans. The Baltic region, Western Russia and Eastern Poland. Central and Western Europe. Italy, with the southern Italians being more distant. In this study, barrier analysis revealed genetic barriers between Finland, Italy and other countries, and interestingly, barriers could also be demonstrated within Finland and Italy. FST was found to correlate considerably with geographic distances ranging from a per-mil 0.0010 currency for neighboring populations to 0.0200-0.0230 for southern Italy and Finland. For comparisons, pairwise FST of non-European samples were as follows: Europeans are Euro-African 0.1530. Europeans are Euro-Chinese 0.1100. Africans are Euro-Chinese 0.1900. A study by Chao Tian in August 2009 extended the analysis of European population genetic structure to include additional Southern European groups and Arab populations from the Near East. This study determined autosomal FST between 18 population groups and concluded that, in general, genetic distances corresponded to geographical relationships with smaller values between population groups with origins in neighboring countries' regions compared with those from very different regions in Europe. Autosomal DNA, Selden used over 5,000 autosomal SNPs. It showed a consistent and reproducible distinction between a Euro-North and a Euro-unregistered trademark and a Euro-South and a Euro-unregistered trademark European population groups. Most individual participants with Southern European ancestry, and Ashkenazi Jews have greater than 85% membership in the Southern population. And most Northern, Western, Central, and Eastern Europeans have greater than 90% in the Northern population group. However, many of the participants in this study were actually American citizens who self-identified with different European ethnicities based on self-reported familial pedigree. A similar study in 2007 using samples exclusively from Europe found that the most important genetic differentiation in Europe occurs on a line from the north to the southeast, with another east-west axis of differentiation across Europe. Its findings were consistent with earlier results based on mtDNA and Y-chromosomal DNA that support the theory that modern Iberians hold the most ancient European genetic ancestry, as well as separating Basques and Sami from other European populations. It suggested that the English and Irish cluster with other Northern and Eastern Europeans such as Germans and Poles, while some Basque and Italian individuals also clustered with Northern Europeans. Despite these stratifications, it noted the unusually high degree of European homogeneity. There is low apparent diversity in Europe, with the entire continent-wide samples only marginally more dispersed than single population samples elsewhere in the world. In 2008, 2 international research teams published analyses of large-scale genotyping of large samples of Europeans, using over 300,000 autosomal SNPs. With the exception of usual isolates such as Basques, Finns and Sardinians, the European population lacked sharp discontinuities as previous studies have found, although there was a discernible south-to-north gradient. Overall, they found only a low level of genetic differentiation between subpopulations, and differences which did exist were characterized by a strong continent-wide correlation between geographic and genetic distance. In addition, they found that diversity was greatest in southern Europe due to a larger effective population size and/or population expansion from southern to northern Europe. The researchers take this observation to imply that genetically, Europeans are not distributed into discrete populations. A study on northeastern populations, published in March 2013, found that Komi peoples formed a pole of genetic diversity that is distinct from other populations. Autosomal genetic distance is based on SNPs. The genetic distance between populations is often measured by fixation index, based on genetic polymorphism data, such as single nucleotide polymorphisms or microsatellites. FST is a special case of F statistics, the concept developed in the 1920s by Saul Wright. FST is simply the correlation of randomly chosen alleles within the same subpopulation relative to that found in the entire population. It is often expressed as the proportion of genetic diversity due to allele frequency differences among populations. The values range from 0 to 1. A 0 value implies that the two populations are panmixes, that they are interbreeding freely. A value of 1 would imply that the two populations are completely separate. The greater the FST value, the greater the genetic distance. Essentially, these low FST values suggest that the majority of genetic variation is at the level of individuals within the same population group. Whilst belonging to a different population group within same Euro unregistered trademark/continent, and even to different racial/continental groups added a much smaller degree of variation. CEU, Utah residents with ancestry from Northern and Western Europe. Apparent migrations into Europe. The prehistory of the European peoples can be traced by the examination of archaeological sites, linguistic studies and by the examination of the DNA of the people who live in Europe or from ancient DNA. The research continues and so theories rise and fall. Although it is possible to track migrations of people across Europe using founder analysis of DNA, most information on these movements comes from archaeology. It is important to note that the colonization of Europe did not occur in discrete migrations, as might appear to be suggested. Rather, the settlement process was complex and likely to have occurred in multiple waves from the east and to have been subsequently obscured by millennia of recurrent gene flow. Paleolithic era Homo neanderthalensis inhabited much of Europe and Western Asia from as far back as 130,000 years ago. They existed in Europe as late as 30,000 years ago. They were eventually replaced by anatomically modern humans, Cro-Magnoid Homo sapiens, who began to appear in Europe c. 40,000 years ago. Given that the two hominid species likely coexisted in Europe, anthropologists have long wondered whether the two interacted. The question was resolved only in 2010, when it was established that Eurasian populations exhibit Neanderthal admixture, estimated at 1.5 2.1% on average. The question now became whether this admixture had taken place in Europe, or rather in the Levant, prior to AMH migration into Europe. There has also been speculation about the inheritance of specific genes from Neanderthals. For example, 1-MAPT locus 17q21.3, which is split into deep genetic lineages H1 and H2. Since the H2 lineage seems restricted to European populations, several authors had argued for inheritance from Neanderthals beginning in 2005. However, the preliminary results from the sequencing of the full Neanderthal genome at that time failed to uncover evidence of interbreeding between Neanderthals and modern humans. By 2010, findings by Svante Park-Currency-Boe, Richard E. Green, and David Reich, comparing the genetic material from the bones of 3 Neanderthals with that from 5 modern humans, did show a relationship between Neanderthals and modern people outside Africa. This suggests that interbreeding occurred in the Middle East. It is thought that modern humans began to colonize Europe during the Upper Paleolithic about 40,000 years ago. Some evidence shows the spread of the Aurignacian culture. From a Y-chromosome perspective, Semino proposed that the large haplogroup A1 is an ancient Eurasiatic marker brought in by Homo sapiens who diffused west into Europe 40 kY ago. Haplogroup I might represent another putative Paleolithic marker whose age has been estimated to 22k year. Whilst it is unique to Europe, it probably arose in descendants of men arriving from the Middle East c. 20 euro 25k year, arising from parent haplogroup IJ. At this time, another Upper Paleolithic culture appears, the Gravettian culture. Thus the genetic data suggests that, at least from the perspective of patrilineal ancestry, modern humans might have taken two colonizing routes, one from the Middle East via the Balkans and another from Central Asia to the north of the Black Sea. It is now believed that the haplogroup A1 is substantially younger. A 2008 study dated the most recent common ancestor of the haplogroup A1 by 18.5k year, and the most recent ancestor of the haplogroup IJ by 38.5k year, suggesting that haplogroup IJ colonists formed the first wave and haplogroup A1 arrived much later. Martin Richards al. found that 40% of extant mtDNA lineages trace back to the Paleolithic migrations. mtDNA haplogroup U5, dated to be 40-50k year, arrived during the first early Upper Paleolithic colonization. Individually, it accounts for 5-15% of total mtDNA lineages. Middle UP movements are marked by the haplogroups HV, I and U4. HV split into pre-V and the larger branch H, both of which spread over Europe, possibly via Gravettian contacts. Haplogroup H accounts for about half the gene lines in Europe, with many subgroups. The above mtDNA lineages or their precursors, are most likely to have arrived into Europe via the Middle East. This contrasts with Y-DNA evidence, whereby some 50% plus of male lineages are characterized by the A1 superfamily, which is of possible Central Asian origin. Ornelas Semino postulates that these differences may be due in part to the apparent more recent molecular age of Y chromosomes relative to other loci, suggesting more rapid replacement of previous Y chromosomes. Gender-based differential migratory demographic behaviors will also influence the observed patterns of mtDNA and Y variation. Last glacial maximum, refugia and recolonization equals. The last glacial maximum started c. 30 Ca BC, at the end of MIS3, leading to a depopulation of northern Europe. According to the classical model, people took refuge in climatic sanctuaries as follows: northern Iberia and southwest France, together making up the Franco-Cantabrian refugium, the Balkans, Ukraine and more generally the northern coast of the Black Sea, Italy. This event decreased the overall genetic diversity in Europe, a result of drift, consistent with an inferred population bottleneck during the last glacial maximum. As the glaciers receded from about 16,000 to 13,000 years ago, Europe began to be slowly repopulated by people from refugia, leaving genetic signatures. Some Y haplogroup I clades appear to have diverged from their parental haplogroup sometime during or shortly after the LGM. Haplogroup I2 is prevalent in the Western Balkans, as well as the rest of southeastern and central Eastern Europe in more moderate frequencies. Its frequency drops rapidly in Central Europe, suggesting that the survivors bearing I2 lineages expanded predominantly through southeastern and central Eastern Europe. Sinioglu sees evidence for the existence of an Anatolian refuge, which also harbored HGO1B1B2. Today, A1B dominates the Y chromosome landscape of Western Europe, including the British Isles, suggesting that there could have been large population composition changes based on migrations after the LGM. Semino, Parsarino and Perisic place the origins of haplogroup A1 within the Ukrainian Ice Age refuge. Its current distribution in Eastern Europe and parts of Scandinavia are in part reflective of a repeopling of Europe from the southern Russian-Ukrainian steppes after the late glacial maximum. From an mtDNA perspective, Richards al. found that the majority of mtDNA diversity in Europe is accounted for by post-glacial re-expansions during the late Upper Paleolithic/Mesolithic. The regional analyses lend some support to the suggestion that much of Western and Central Europe was repopulated largely from the southwest when the climate improved. The lineages involved include much of the most common haplogroup, H, as well as much of K, T, W, and X. The study could not determine whether there were new migrations of mtDNA lineages from the Near East during this period. A significant input was deemed unlikely. The alternative model of more refugees was discussed by Bilton et al. Neolithic migrations equals a big decline in genetic variation that has long been recognized in Europe seems to show important dispersals from the direction of the Middle East. This has often been linked to the spread of farming technology during the Neolithic, which has been argued to be one of the most important periods in determining modern European genetic diversity. The Neolithic started with the introduction of farming, beginning in SE Europe approximately 7000 to 3000 BC, and extending into NW Europe between 4500 to 1700 BC. During this era, the Neolithic Revolution led to drastic economic as well as socio-cultural changes in Europe, and this is also thought to have had a big effect on Europe's genetic diversity, especially concerning genetic lineages entering Europe from the Middle East and the Balkans. There were several phases of this period. In a late European Mesolithic prelude to the Neolithic, it appears that Near Eastern peoples from areas that already had farming, and who also had seafaring technology, had a transient presence in Greece, for example at Franchvi Cave. There is consensus that agricultural technology and the main breeds of animals and plants which are farmed entered Europe from somewhere in the area of the Fertile Crescent and specifically the Levant region from the Sinai to southern Anatolia. A later stage of the Neolithic, the so-called Pottery Neolithic, saw an introduction of pottery into the Levant, Balkans and southern Italy, and this may have also been a period of cultural transfer from the Levant into the Balkans. Spread of Neolithic technology. An important issue regarding the genetic impact of Neolithic technologies in Europe is the manner by which they were transferred into Europe. Whether farming was introduced by a significant migration of farmers from the Near East or a cultural diffusion, or a combination of the two. Secondarily, population geneticists have tried to clarify whether any genetic signatures of Near Eastern origin correspond to the expansion routes postulated by the archaeological evidence. Martin Richards estimated that only 11% of European mtDNA is due to immigration in this period, suggesting that farming was spread primarily due to being adopted by indigenous Mesolithic populations, rather than due to immigration from Near East. Gene flow from SE to NW Europe seems to have continued in the Neolithic, the percentage significantly declining towards the British Isles. Classical genetics also suggested that the largest admixture to the European Paleolithic Mesolithic stock was due to the Neolithic revolution of the 7th to 5th millennia BC. Three main mtDNA gene groups have been identified as contributing Neolithic entrants into Europe, J, T1 and U3. With others, they amount up to around 20% of the gene pool. In 2000, Semino's study on Y-DNA revealed the presence of haplotypes belonging to the large clade E1b1b1. These were predominantly found in the southern Balkans, southern Italy and parts of Iberia. Semino connected this pattern, along with J haplogroup subclades, to be the Y-DNA component of Cavalli-Sforza's Neolithic demic diffusion of farmers from the Near East. Rosser et al. Rather saw it as a North African component in European genealogy, although they did not propose a timing and mechanism to account for it. Underhill and Kivisild also described E1b1b as representing a late Pleistocene migration from Africa to Europe over the Sinai Peninsula in Egypt, evidence for which does not show up in mitochondrial DNA. Concerning timing the distribution and diversity of E13, however, Battaglia et al. Proposed an earlier movement whereby the EM78 lineage ancestral to all modern EV13 men moved rapidly out of a southern Egyptian homeland and arrived in Europe with only Mesolithic technologies. They then suggest that the EV13 subclade of EM78 only expanded subsequently as native Balkan foragers cum farmers adopted Neolithic technologies from the Near East. They proposed that the first major dispersal of EV13 from the Balkans may have been in the direction of the Adriatic Sea, with the Neolithic impressed where culture often referred to as impressor or cardial. Peria et al. Rather proposed that the main route of EV13 spread was along the Vardar-Morava-Danube river highway system. In contrast to Battaglia, Cruciani et al. tentatively suggested a different point where the V13 mutation happened on its way from Egypt to the Balkans via the Middle East, and a later dispersal time. The authors proposed that the V13 mutation first appeared in Western Asia, where it is found in low but significant frequencies, whence it entered the Balkans sometime after 11K year. It later experienced a rapid dispersal which he dated to c. 5,300 years ago in Europe, coinciding with the Balkan Bronze Age. Like Peria KL. They consider that the dispersion of the EV13 and JM12 haplogroups seems to have mainly followed the river waterways connecting the southern Balkans to north-central Europe. More recently, Lake Anale announced that a 7,000-year-old skeleton in a Neolithic context in a Spanish funeral cave was an EV13 man. Using 7 STR markers, this specimen was identified as being similar to modern individuals tested in Albania, Bosnia, Greece, Corsica, and Provence. The authors therefore proposed that, whether or not the modern distribution of EV13 of today is a result of more recent events, EV13 was already in Europe within the Neolithic, carried by early farmers from the eastern Mediterranean to the western Mediterranean, much earlier than the Bronze Age. This supports the proposals of Battaglia al. Rather than Cruciani al, at least concerning earliest European dispersals, but EV13 may have dispersed more than once. Even more recent than the Bronze Age, it has also been proposed that modern EV13's modern distribution in Europe is at least partly caused by Roman-era movements of people. After an initial focus upon E1b1b as a Neolithic marker, a more recent study in January 2010 looked at Y haplogroup A1b1b, which is much more common in Western Europe. Mark Jobling said, "We focused on the commonest Y-chromosome lineage in Europe, carried by about 110 million men. It follows a gradient from southeast to northwest, reaching almost 100% frequency in Ireland. We looked at how the lineage is distributed, how diverse it is in different parts of Europe, and how old it is. The results suggested that the lineage A1b1b2, like E1b1b or J lineages, spread together with farming from the Near East." Dr. Patricia Balaresk added, "In total, this means that more than 80% of European Y chromosomes descend from incoming farmers. In contrast, most maternal genetic lineages seem to descend from hunter-gatherers. To us, this suggests a reproductive advantage for farming males over indigenous hunter-gatherer males during the switch from hunting and gathering to farming." A more recent article concerning A1b made the counterclaim that the data are still controversial and the analyses so far performed are prone to a number of biases, and proposed that the data are best explained by an earlier, pre-Neolithic dispersal of haplogroups from a common ancestral gene pool. One hypothesis The Anatolian hypothesis suggests an origin of the Indo-Europeans in Anatolia with an expansion due to the Neolithic Revolution. Bronze and Iron Age migrations The Bronze Age saw the development of long-distance trading networks, particularly along the Atlantic coast and in the Danube Valley. There was migration from Norway to Orkney and Shetland in this period. There was also migration from Germany to eastern England. Martin Richards estimated that there was about 4% mtDNA immigration to Europe in the Bronze Age. Another theory about the origin of the Indo-European language centers around a hypothetical Proto-Indo-European people, who traced in the Kurgan hypothesis, to north of the Black and Caspian Seas at about 4500 BC. They domesticated the horse and possibly invented the wheel, and are considered to have spread their culture and genes across Europe. The Y haplogroup A1A is a proposed marker of these Kurgan genes, as is the Y haplogroup A1B. Although these haplogroups as a whole may be much older than the language family. The rate of their physical expansion would have declined at the western edge of the steppe, but carriers of the A1A haplogroup are present in substantial numbers as far west as Germany. The Kurgan culture and language went farther, carried by the A1B haplogroup, and eventually replacing most cultures and languages all the way to the Atlantic. During the Iron Age, Celts are recorded as having moved from Gaul into Italy, Eastern Europe and Anatolia. The relationship between the Celts of Gaul and Spain is unclear as any migration occurred before records exist. In the far north, carriers of the Y haplogroup N arrived to Europe from Siberia, eventually expanding as far as Finland, though the specific timing of their arrival is uncertain. The most common North European subclade N1C1 is estimated to be around 8,000 years old. There is evidence of human settlement in Finland dating back to 8500 BCE, linked with Kunda culture and its putative ancestor Swiderian culture, but the latter is thought to have European origin. The geographical spread of haplogroup N in Europe is well aligned with the Peter-Euro-Khmer culture, whose emergence is commonly dated c. 4200 BCE, and with the distribution of Uralic languages. Mitochondrial DNA studies of Sami people, haplogroup U5 are consistent with multiple migrations to Scandinavia from Volga-Ural region, starting 6,000 to 7,000 years before present. The relationship between roles of European and Asian colonists in the prehistory of Finland is a point of some contention, and some scholars insist that Finns are predominantly Eastern European and made up of people who trekked north from the Ukrainian refuge during the Ice Age. Farther east, the issue is less contentious. Haplogroup N carriers account for a significant part of all non-Slavic ethnic groups in northern Russia, including 37% of Karelians, 35% of Komi people, 67% of Mari people, as many as 98% of Nenets people, 94% of Anganizans, and 86% to 94% of Yakuts. Roman and post-Roman period during the period of the Roman Empire, historical sources show that there were many movements of people around Europe, both within and outside the empire. Historic sources sometimes cite instances of genocide incited by the Romans upon rebellious provincial tribes. If this did in fact occur, it would have been limited given that modern populations show considerable genetic continuity in their respective regions. The process of Romanization appears to have been accomplished by the colonization of provinces by a few Latin-speaking administrators, military personnel, settled veterans, and private citizens who emanated from the empire's various regions. They served as a nucleus for the acculturation of local notables. Given their small numbers and varied origins, Romanization does not appear to have left distinct genetic signatures in Europe. Indeed, Romance-speaking populations in the Balkans, like Romanians, Aromanians, Moldovans, etc. have been found to genetically resemble neighboring Greek and South Slavic-speaking peoples rather than modern Italians, proving that they were genetically speaking, mainly through I2A2M423 and E1B1B1, V13 haplogroups native to this area. Stephen Bird has speculated that E1B1B1A was spread during the Roman era through Thracian and Dacian populations from the Balkans into the rest of Europe. Concerning the late Roman period of Germanic var paragraph Lkewanderung, some suggestions have been made, at least for Britain, with Y haplogroup I1A being associated with Anglo-Saxon immigration in eastern England, and A1A being associated with Norse immigration in northern Scotland. See also Y-DNA haplogroups by groups in Europe, African admixture in Europe, European ethnic groups, by area, archaeogenetics of the Near East, genetic history of indigenous peoples of the Americas, genetics and archaeogenetics of South Asia, general, archaeogenetics, timeline of human evolution, human genetic variation, population genetics. References. References Bibliography External links, Atlas of the Human Journey, World Haplogroups Maps, Genetic Structure of Human Populations. Haplogroup A1b,